All right. So today, Djordje and I are going to present our paper that we read titled Lamin B1 Loss Promotes Lung Cancer Development and Metastasis by Epigenetic Depression of RET. It's kind of a long title, but
Uh, can I just say, I'm excited. Are we going to be in the nucleus?
yeah, we're getting to the
Yeah,
nucleus.
hell yeah. You know, the nucleus I feel like doesn't get enough play, so I'm excited.
That's what I'm saying. So just our little personal connection here is that we've been stuck in quarantine for quite a bit, which leads to a lot of time to watch Netflix. And one program featured on Netflix that Djordje and I both enjoy is Breaking Bad. And um, the protagonist, Walter White, comes down with lung cancer, and maybe we were like, all right, we need a disease. What possible mechanics are involved here with lung cancer? So which led us to discover this paper. But just some background, what we already know from class, we've seen sort of this mechanical regulation of transcription uh, from mechanical regulation of transcription. We read that one paper. They talked about uh, lamin A and C, I believe. I don't think they get into lamin B1, which is the one we're talking about. But yeah, we see that we know that gene transcription can be mechanically regulated. So some background, lung cancer, it's a leading cause of cancer-related death worldwide, and one of its characteristics is alterations in the nuclear structure. So we um we see changes in the number of nuclei, and one specific is we see this polymorphic nuclear envelope. And so basically it's not round like the one in the left, the nucleus in the left. It's more like the one on the right, like the metastatic cells. They're kind of lumpy and misshapen, and um and
That's,
so
can I just have a question? So that's in cancer cells?
yes, lung
Lung
cancer cells.
cancer. Yeah, they have these messed up looking nuclei
um
in like most of the cells. Is that what it is? No.
yes, I think so. I know actually the cells that are in this picture are actually prostate cancer cells. So maybe it's not just cells for lung cancer, but
Well,
maybe cells in other
prostate and lung are both very like glandular and ductal, so they share some similarities for sure.
yeah.
Both pretty lethal cancers too.
hmm Um yeah, so they kind of are saying, okay, so maybe this bad shape is coming from uh changes in the nuclear lamina, which, like, I think we've learned this before, the nuclear lamina is just meshwork of intermediate filaments and lamins, and which is what we're going to talk about is the lamins. And the lamins support the structure of the nuclear envelope and provide anchorage sites for chromatin. And they have seen in the past, prior to this paper being released. So this paper came out in 2019, so it's very recent. So prior to this paper, they knew that changes in expression to lamins have been linked to cancer phenotypes, but the direct function of the lamins in cancer was unclear. So that's what they might get into in this paper. And lastly, just some proteins that we're going to talk about in the paper. Lamin B1 is, the lamin that they focus on, and the genes associated with it um, are usually the ones that uh, methylate different, or yeah, lead to methylation of histones. And what methylates those histones is the PRC2, the polycomb repressive complex 2, and it has the proteins EZH1 and 2, which catalyze the methylation of H3 on the lysine residue. And lastly, they talk about RET, which is a receptor tyrosine kinase, which stimulates a signaling pathway that contributes to cancer progression.
And just to clarify, I think that EZH1 and 2 are just domains of the PRC2 complex. They're not separate proteins, but yeah. And then going forward to their actual.
Can we go back to that last slide just so I can look at it? Oh, sorry, it's kind of loud. Oh yeah, okay. So we have basically lamin B1s at the nuclear kind of envelope. And then the
Yep.
PRC and EZH. Does EZH connect to the nuclear envelope at all? I can't remember.
I don't.
I feel like this.
I think EZH connects to whatever it's going to attach a histone mark to.
Okay.
Or
Yeah.
methylation.
So those are all at the DNA then. And then the receptor tyrosine kinase, that's out at the cells.
That's the plasma membrane.
Yeah.
Plasma membrane. Yep.
Yeah. Okay. So these three things are in the nucleus that we're talking about, and then something out the plasma membrane. Cool. Thanks.
Yep.
Yeah.
And then moving on to their experiments. In this first figure on the left, along the x-axis, they have the first lane is just the normal wild type cell, and then they have increasing grades of cancer cells. And then along the y-axis, they have like a Lamin B1 score. And this is just like a quantification of an immunostaining experiment that they did. So this just shows that cancerous cells have lower Lamin B1 levels compared to a wild type control. And then in the next experiment, they silenced Lamin B1 with CRISPR and siRNA, and they monitored other protein levels and found that in Lamin B1 knockdown cells, there was an upregulation of mesenchymal markers such as N-cadherin, fibronectin, and vimentin. And then this paper is like 19 pages, and they have like 20 different experiments. So they ran other experiments that showed that Lamin B1 knockdown cells have a greater migratory capacity and form larger colonies compared to wild type cells. And then in the rightmost figure here, they ran this experiment in vivo with mice. So they injected mice with Lamin B1 knockdown cells, and then they monitored different variables. And this is just like one of the panels that that is in the paper that we chose to include. So you can see that Lamin B1 knockdown cells have bigger tumors. So on the left two lanes or panels show kind of like a normal wild type mouse's lungs. And then on the right side, you can kind of see that it's all messed up. So when you knock down Lamin B1, it promotes tumor growth. Moving on.
No, this is all good. Not yet. I got questions. You're doing a great job. I have some questions about like the experimental setups here, just so I can understand better. So they're like over on the left here, they have like cancer cells in a dish that they're playing with, right?
So on the left is just 1 second.
Yeah, the leftmost figure is they. So what they get this data from is this is figure C. So figure A and B are actually they look like light microscope images, and they stand for anti-Lamin B1.
Okay, so but they're just cells like in a dish.
Yeah, it's just a quantification. Yeah. Just cells in a dish.
So that's also coming from B, right? That's B.
Well.
Yeah, same thing.
Are the images. Okay, and then in H, so are these like a tumor line of mice that automatically make tumors?
No, it says that they inject control and Lamin knockdown cells intravenously into nude mice, and then they
so they're injecting tumor cells into the lungs of mice and then looking at them. I see. Cool. Thank you. That makes sense.
Yeah. I don't know if they're specifically injecting tumor cells
or cancer cells.
Yeah.
Or I mean,
it just says that they inject Lamin B1 knockdown cells.
Oh yeah, so they're injecting just control cells, and they don't make any tumors. That's what I was curious about. I was like, are these making tumors at all? And they're not.
They're not. But then when you inject Lamin B1 knockdown, they are.
Yeah, they make a ton. So just by knocking down, okay, that makes sense because you're knocking down the lamin is protecting the nucleus, and so if you knock that down, you take away the nucleus's structure. Your DNA is just going to be getting banged around, bro, anytime you're moving. So you're going to get mutations and cancers. Cool. All right.
Yeah. And then later, we're going to go over it exactly how Lamin B1 regulates this process.
That's sweet. Yeah. I'm excited.
And so then on the next slide, they wanted to investigate the mechanisms by which Lamin B1 mediates EMT and metastasis. And so they performed RNA sequencing with Lamin B1 knockdown and wild type cells to see kind of what the differences between the two are. And they found that this specific protein, RET, was upregulated in the knockdown cells. And so that's what this first figure on the left kind of shows. It just shows that the RET is the control, and you can see low levels of RET. And then as soon as you look at the knockdown, you have super high levels of RET expression. So RET is upregulated in Lamin B1 knockdown cells. And then, so they wanted to further investigate RET. And so they either silenced it or overexpressed it.
RET was a tyrosine kinase, is that correct?
RET is the tyrosine kinase, yeah.
Okay, thank you.
And so then they either silenced it or overexpressed it and monitor other protein levels. And that's this middle figure. And so way on the right side, you can see RET overexpression. And then you can see that when you have RET overexpression, you have like increased levels of NCAD-herin and other mesenchymal markers. But then when you knock down RET, so in the third and fourth lanes in the RET knockdown, you see a decrease of these mesenchymal markers. And so they also ran these experiments then in vivo again with mice, which is the rightmost figure. And they found that RET knockdown inhibited the increased tumor growth and metastatic ability of Lamin B1 knockdown cells. And so I'll take us through this figure. So if you look at the second lane for each of these panels, you can see that there's a huge jump in the metastatic area. So it's basically saying that when you knock down Lamin B1, you're going to have a lot of tumor growth. And then when you look at the fourth lane, which is both Lamin B1 knockdown and RET knockdown, you can see that this tumor growth is kind of brought back to a control level, which kind of shows that RET knockdown can bring it back, if that makes sense.
So I have a question.
Sure.
It shows that RET knockdown decreases tumor growth and LMNB knockdown increases tumor growth. Are those two effects, like, do they show in some way that those two effects are actually related, or is it just they have opposite effects that kind of complement each other?
So what happens, I think they might show this later, but what they end up saying is that Lamin B1, like the PRC2 complex, ends up that works downstream of Lamin B1, ends up methylating the histones that control expression of RET. So like so what ends up happening is that without Lamin B1, you could see in the one labeled D, that Lamin B1 RET is upregulated because RET levels are higher because it's not regulated by this methylation controlled by Lamin B1. And then RET overexpression, they'll go on to say, leads to tumor progression because of the downstream signaling from the receptor tyrosine kinase.
Yeah, so they have a few more experiments that kind of show that Lamin B1 regulates RET through methylation. So that's kind of the link between the two.
I kind of want to jump in here. Are people familiar with the idea that like your cells in your body kind of have like a timer? They kind of have like a burning wick for how long they live. And there's kind of different methods that cells use to sort of kill themselves and to have that wick. And one is like telomeres, and there's other things like adding a bunch of sugars to proteins and things like that. Are people familiar with that idea?
Yeah, but I don't know too much about it, but I've definitely.
Yeah, no, I'm just saying in general, just like
exists. Yeah,
generally that.
Mm-hmm. Yeah,
I know that it exists.
Yeah, and like that's like one of the key like important things that like we got from Henrietta Lacks, the person who's like cancer cells were just kind of used without their knowledge or the family's knowledge, is those cells were immortalized because they're cancer cells. that basically a cancer cell is a cell that hijacks a way to turn off that wick so that it just keeps burning forever, and then it can do whatever it wants. And then it has its own evolutionary path where it gets to like evolve over time and get new traits. And so like this almost seems like this interplay right here, when we have a messed up neuron that's got its lamin deleted, then the cell has RET genes kind of like locked away by lamin. So if lamin's gone, now we have all these extra RET genes being opened up, or just, you know, one RET gene being opened up or whatever, two, whatever it is. So that then we make more RET. So then basically kind of make it almost want to start to die, or does it want to become a cancer cell? You know what I'm saying?
To become a, yeah, what RET promotes
It promotes proliferation
and proliferation, so
it's like
promotes cancer qualities.
Yeah, so like I'm curious.
Cancerous qualities.
It's promoting more cancerous qualities. So this would be like an oncogene. So when it gets turned on,
they call it an oncogene in the paper.
Yeah,
too.
and so then, it's really interesting. So then we're not even getting into what I was just talking about, this whole wick thing. We're just jumping right into cancer. And so now that this cell is able to go and divide and keep going. Yeah, so what is it about? Is there anything specific about these cells that allows them to then just continually divide? Why don't they eventually die off? Like, why does RET keep them like stem-like so they keep dividing? Do we know anything about that?
Well, aren't they starting with cancer lines in the first place?
Are they starting with cancer lines?
I thought they were.
Okay, so
yeah,
then,
I'm pretty sure before they start all of these modifications, the cells are already cancerous, and they're just looking at the growth of already cancerous cells.
Yeah, so now what happens in a control cell? But I think, but no, weren't they doing that? Sorry, I'm into this.
Yeah, I think that in their like in vivo experiments, I don't think that the controls are cancerous cells.
Yeah, so then they're just basically making them cancerous by deleting lamin.
Yeah.
Okay,
I think
then
so.
I must have misunderstood.
Yeah, that's fine. Yeah, I'm not sure which way it is, but I think that's a really interesting question. We can move on from that, but I think there's a lot that we don't know about, those like tiny little interplays between like making something cancerous versus like keeping its natural shutdown clocks intact. Because it doesn't really matter if you add a bunch of cancerous genes if you still have those die after 70 divisions. Because if that tumor is just going to kill itself. It's only when you get that combination of them where it really gets icky, where they can continually live forever, and they're on a tirade going bananas.
I remembered reading this, so I looked at the paper. It says RET signaling induces neural crest cell migration by modulating actin dynamics and inducing lamellipodia formation.
Yeah, so that's interesting.
RET does.
I mean, that's why I got into neural crest cell migration, because it's so close to cancer migration, but it's just normally happening. So you can repeatedly watch it over and over again without having to do any like modifications and making things cancerous. Okay, cool. Let's move on. Thank you. That was a great little dig in there for me.
So then with their next set of experiments, they looked specifically at lung cancer cells, which is this figure A on the left, and they stained for RET. And so in the leftmost panel, you see a normal healthy lung cell or healthy lung. And then in the next three panels, you can see increased levels of RET. So this is kind of just like solidifying the results that in cancer cells, there will be increased levels of RET. And then in the right figure, it basically just shows the inverse relationship between Lamin B1 and RET. So like the more RET you have, the less Lamin B1. The more Lamin B1, the less RET, or the RET score is. So this supports a link between Lamin B1 loss and RET upregulation in lung cancer patients. The researchers then wanted to investigate how exactly Lamin B1 might silence genes such as RET in wild type cells. And so in the figure on the left, they show that basically when you look at the control cells, EZH1 and 2 were not recruited to the RET, if you look at the knockdown cells, you see that EZH1 and 2 were not recruited to the RET promoter, whereas in the control cells, there is a strong association of EZH1 and 2 to the promoter region of RET. And then, so EZH1 and 2, like we talked about, add those repressive histone marks. And so then this is, if you look at the left figure, in the control, if you're adding those repressive histone marks to RET in the controls, then RET won't be expressed, and the opposite is true for the knockdown. And then figure E is kind of just showing the same thing. If you look at the control, which is the red line, you see a spike in methylation levels, or in methylation marks. And then if you look at the knockdown, you see that the spike is now absent, which means that whatever genes were being suppressed by these methylation marks might now be active.
Yeah, so basically what this paper found, I thought it was structured very well, like the way they kind of outlined their figures. So the first thing they showed, cancer cells have lower Lamin B1 levels. So then they examined that. So they showed that Lamin B1 knockdown promoted EMT, cell migration, tumor growth, and metastasis. So they thought, okay, what exactly is Lamin B1 knockdown causing? So they did a genome-wide screen, and not genome-wide, they did a qPCR. They did some sort of thing. They did something that came up with a RET. And they saw that RET upregulation promotes, Lamin B1 causes RET upregulation, and RET upregulation promotes EMT phenotypes. And they found, okay, so why is Lamin B1 knockdown causing RET upregulation? Well, Lamin B1 recruits PRC2, which normally represses RET via methylation. Specifically EZH1 and 2 cause that methylation. And they basically came up, Lamin B1 knockdown causes RET activation, and the downstream effects of RET activation cause tumor formation.
They were nice enough at the end of their paper to include a model. Can you guys hear me?
Mm-hmm.
Hello?
Yeah.
I can hear
Yeah,
you, but it cut out a little bit for me.
Yeah, it said that I had bad internet quality for a sec, but
yeah, it's a little,
it should be
but
good
it's
now.
okay. It's okay.
Okay. So yeah, and they were nice enough to include a model at the end of their paper. I think that we thought it did a pretty good job of summarizing the results that they found from their experiments. So on the left, you can see kind of like a normal cell with high Lamin B1 levels. Lamin B1 is that those little purple lines kind of on the inside of that membrane. And you can see that when Lamin B1 is there, EZH1 and 2 is able to add those little red triangles, which signify the methylation marks, and that RET gene is turned off and not expressed. And then on the right side, when you have low Lamin B1 levels, EZH1 and 2 isn't there to add those red triangles, and so then RNA polymerase can come onto the RET gene, promote transcription. You can make that RET receptor. The tyrosine kinase can go on and phosphorylate whatever it needs to, which leads to EMT and metastasis. Once again, we thought that it was a pretty good model. All the parts are labeled, color scheme's good, red off, green on. We thought maybe it would be helpful to show polymerase floating around and unable to attach on the left side of the model, just to kind of hammer home that point a little more. But other than that, we thought it was good.
I was thinking it might be nice to have because they're gonna have more RET up there, so having it be overexpressed, having multiple copies would be nice.
Yeah. That too.
Yeah,
and also
that's
labeling that the top line is a plasma membrane.
Yeah, that's
That threw
be
me
nice.
off a little bit at
there's,
first.
And
there's
the
like 3
nucleus,
little
too.
lines.
Yeah.
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 I mean, it does look it looks a little hazardly put together, but it is pretty. It's,
They
I
could have,
like,
it
I
gets
love
the
it.
point across, but they
Yeah.
could have easily made like a whole cell, you know, with like,
I like it. I like it a lot, though. It's very good.
hmm
As far as models go, they're usually way too complicated. This one's nice and simple.
hmm So we came up with a little bit of an analogy now to show kind of how Lamin B1 is like our modern day situation.
It's topical.
So it is topical. So Lamin B1, if there's a lot of it, if there's a lot of it, then there's no cell migration, no like no metastasis, no like
moving
much,
around,
no moving around.
proliferation.
You can't leave your house. But if Lamin B1 kind of goes away and there's less of it, you got a little RET key that will unlock your door for metastasis. Not to imply anyone here is a cancer, but you know, if you can leave your house just like a cancer cell can move around and proliferate and all that stuff.
We can apply that humans are a cancer to the planet. We could do it.
Yeah.
Heck, there's already plenty of comparisons to viruses. Throw cancer on the pile too.
Yeah.
I mean, we're more like cancer because we migrate. You know, we live on our own. We can keep going and replicating. You know, virus depend. I mean, we're viral in a way. We're a virus. Okay, no, I'll shut up.
I mean, we do require eating up everything around us to continue living.
True. Yeah. So yeah, that's basically our model. So
I especially like the little viruses.
oh, thank you. It was my
that was Lucas's art.
a little Adobe Illustrator skills.
They're fantastic. And this reminds me of like Salad Fingers, this house or something. Like the art style. It's so like flash animation. I like it.
Thank you.
And then for our discussion assignment, we can just do it as a group unless you want to put us into break rooms. As a researcher, what might your next steps or experiments be after reading this paper for gaining a better understanding of this mechanism, or generating a potential treatment for lung cancer?
Let's just do it as a group. One thing I'm thinking about is like, can we just do some easy way to do histone modification as a medicine? Histone modification medicine seems like the next thing now to me after like this class, and like because then you could cut out the force stuff, you can cut out the chemistry stuff. If we could just get to the DNA and modify the histones, then you don't need to edit. You don't need to edit the gene even. You just turn it on or off. You know.
Would that actually work, or would the cell recognize that it's modified strangely and reverse it unless you undo the mechanism that leads to that modification state in the first place?
Yeah, we just got to be smarter than the cells.
Yeah.
I think it's sometimes we are, sometimes we're not. That's for sure. But yeah, that's a great pitfall that you brought up there, Aaron. Yeah, that's, if I was writing my grant and I didn't mention that and you were reviewing it, you'd have me checkmate right there.
Yeah.
Yeah, so that's a good point.
So we have a whole slide after this, just really quickly, where we're going to talk about like the medical relevance.
Yeah.
And so that's a potential thing to look at, at the histone marks and see if you can
right, because if you could just shut off the RET expression here, then you don't have this problem, right? So if we could just somehow like basically CRISPR histone modify, does that mechanism exist?
Yeah,
can we figure that out? Yeah, I'm not sure. It's cool.
And then if nobody has anything for like the part A, like gaining a better understanding of this mechanism, after I first read this paper, I thought that it was really interesting that they didn't try to run any experiments to figure out what leads to Lamin B1 downregulation in the first place. Because that's like the root of all these problems, right? Like Lamin B1 gets downregulated, so all these other things happen.
Yeah.
And they say that there's not much data on it, but that there may be some kind of hypermethylation marks at the Lamin B1 promoter that lead to these. So I think that that would be like next steps to kind of run experiments on to gain a better understanding of the bigger picture of like how this starts in the first place.
Yeah, that is interesting. Did they think it had anything to do with force? Did they talk at all about that? Like what?
I don't think so, but
it could because I know excessive force on nuclei and making cells crawl through like little thin channels can. Go ahead.
Yeah, that's kind of what I was thinking of, because like when I was talking about the paper we read earlier about how like there's mechanical regulation of gene transcription, maybe like it has something to do with force, like some sort of mechanics that'll like affect
like mechanical breakdown of lamin. So it's not even a gene expression thing. It's like by like just like if you take I've got this like if I take this foil and I just go like this to a shitload, it gets softer over time, right? Because it becomes more malleable.
Yeah.
So then eventually the cell could notice that this thing's busted. Let's break that fucker down. So if you're just mechanically busting up your lamins, and then the cell's saying like, we got to break this down, but you can't make enough to replenish it, then you've got this issue, right? So that could be one way.
It could be.
I don't know if that's definitely happening, but it's a possibility.
No, it's a good thought. And then kind of we just have one last slide.
Yeah. Cool. So like, we kind of just we talked about this a little bit before, but like also what the researchers were talking about in their discussion was perhaps like they can look at the benefit, or they have looked at the benefit of RET inhibitors in hold on, the people's faces are taking, I can't read this slide. The benefit of RET inhibitors have been shown in several clinical trials. So like they've been exploring different RET inhibitors, P38 signalings, and they've kind of seen it reverse this phenotype. But again, like maybe that's kind of downstream of the whole Lamin B1 knockdown, and perhaps we could look at what would happen if you like what would happen to knock down Lamin B1, like kind of study that in the future, or like maybe there'll be drugs to overexpress Lamin B1, or or these things.
Yeah, and like, then they also this paper is. Go ahead.
I was just gonna say, like thinking back onto like that little idea, like thinking about like overexpressing Lamin B1, or like how do we just like keep the Lamin B1 that we have intact and there at the nuclear membrane, like how do we just preserve and prevent breakdown, ubiquitination, or whatever it is, I don't know.
And then also just like as Lucas said, this paper is really new, so these clinical studies are like even newer, and so there's like I like looked at the links that for the references, and they're kind of like ongoing clinical trials. So I think they've shown like some potential, but still needs to be more work done with them. Yeah, that's all.
All right. Do we have any questions?
Questions? If not, then thank you for listening.
Thank you.
That was cool. I learned a lot. I thought that tied in very nicely with the nuclear paper that we did. Used kind of what we knew and had some of the same proteins, but gave us a new playground in there. So thank you for expanding that world for me. The nucleus is someplace that I never got to spend a lot of time on researching, but it's basically a cell within a cell. So it's a fascinating little world to me. So thank you for teaching me more about it.